Electrophoresis is a process that uses an electrical current to move negatively charged pieces of DNA through a piece of gel. Gels have been prepared for you that have small wells where you can insert segments of DNA in a mixture. To do this, you'll use a micropipette. Micropipettes are different and much more precise from the plastic transfer pipettes you're used to using. You'll notice there's a series of numbers here. This micropipette can be used to measure from 10 microliters to 100 microliters. The first number at the top would read 0, and then 1, and then 3, meaning the total volume of this is 13 microliters. To use the micropipette, begin by placing a tip from the tip box, which has been sterilized so it's sealed with tape, on the micropipette. To do this, simply hold the micropipette and insert it into a tip. Be sure to close the lid to keep the remaining tips sterile. You'll practice first with a loading gel. Hold the micropipette in your hand, press down on the top so you can see it depresses, and you will take up precisely 13 microliters. Be sure to hand the loading tube sample to your partner to cap it so it does not spill. In order to load the gel properly, balance your arm and insert the tip of the micropipette into the well and depress, pulling up. Be careful not to puncture the gel or move the gel from side to side. When you're finished with your tip, place it into the 10% bleach waste beaker solution. To remove the tip, push down on the white lever and eject the tip. You'll do this with the remaining samples A, B, C, and D, which represent plasmids that have been cut with different restriction enzymes. Recall that a plasmid is a circular piece of bacterial DNA. These have been digested with different restriction enzymes. Be careful to note which letter represents which sample because not all samples A will be yellow. Because there's such a small volume, as you can see, you can use a microcentrifuge to spin the sample down to the bottom of the test tube so that you can extract it more easily. All of your samples have been loaded into the gel with the wells on the black side running to the red side because DNA is a negatively charged particle and we want it to run to red. Place the lid on the gel box. Once the lid is slid into place, you may turn on the power supply. You'll be running your gel at 120 volts. You can adjust if it's not already set at the proper voltage by turning this dial on the gel box. You'll know that the gel is running properly when you see small bubbles moving up the side of the gel box. These bubbles will indicate that an electrical current is moving through the gel. After about 20 minutes, you'll see that your loading die has moved down your gel. You may not be able to see the smaller fragments, but we can stain the gel and then see our results.